All right. Welcome, everyone. We're going to go ahead and get started with our Medical Scholars Seminar tonight. Before I hand the mic over to Dr. Friedlander to introduce our speaker, I wanted to let you know that we are actively looking for nominees for next year's Medical Scholars Seminar Series. So if you know of or have worked with individuals who are practicing physicians and exemplify our identities of scientist, physician, master adaptive learner, and system citizen, please consider nominating them. It allows us to host somebody that you've maybe worked with and show them what we're doing here at VTC. So we love to have individuals who are really demonstrating in their day-to-day practice how the spirit of inquiry is informing both how they care for patients and how that informs the research that they do. So I'm going to be looking forward to hearing tonight's seminar, as well as our last seminar for this academic year, which will be on May 5th. And that'll be Dr. Nicole Kalakos from Duke. So be marking that on your calendar. And without further ado, I'm going to ask Dr. Mike Friedlander to introduce tonight's speaker.
Thank you, Leslie. Hi, everybody back there. Hello. And up front, too. So it really is a pleasure to introduce your speaker tonight, who's a personal friend and colleague of mine of many years, Dr. Lisa Guy-Woodford. Dr. Guy-Woodford is a professor of pediatrics in nephrology at the University of Pennsylvania School of Medicine. And also she serves as the Presidential Endowed Chair for Clinical and Translational Science at the Children's Hospital of Philadelphia. By way of quick background, Dr. Guy-Woodford did her undergraduate work at the College of the Holy Cross, where I think she studied biology, if I remember correctly, and then did her medical degree at Harvard University. From there, she completed a residency in pediatrics, followed by a fellowship in pediatric nephrology at the Children's Hospital of Boston, where she studied, she went to study genetics at the Jackson Lab up in Maine, for those of you who have been there. She completed the residency in pediatrics, followed by a fellowship in nephrology, as I said. She then joined the faculty at the Department of Medicine at the University of Alabama, Birmingham, where I got to know her a few years ago, UAB. And there she became professor and endowed chair in medical education as well. She also served as the vice chair of genetics at UAB, where she also served as director of the Center for Clinical and Translational Science and director of the Inherited Renal Disorders Clinic. So as you can see, in addition to her incredible pedigree and credentials as a clinician and a scientist, she was very well recognized early on in her career as a leader and somebody who was able to take the helm and move programs in a very, very positive direction. In 2012, Dr. Guy-Woodford moved to the Children's National Hospital in Washington, D.C. as the Hudson Professor of Pediatrics and Director of the Inherited and Polycystic Kidney Disease Program. And she served there as Associate Vice President for Clinical and Translational Research at the Children's National Hospital partner, George Washington University. I should add, at that time, she became an adjunct professor at the then Virginia Tech Carilion Research Institute, now known as the Fralin Biomedical Research Institute, a position she has held since that time. So we're very proud that she has been one of us since the earliest days. 3 years ago, she moved to her current position at Penn and CHOP, where she also serves as the Associate Chief Scientific Officer for Clinical and Translational Science and as the site PI for the Institute for Translational Translational Medicine and Therapeutics. I should add that she, in addition to her adjunct appointment here, she has received numerous forms of recognition for her work. I won't go through them all, but just mention a couple quickly. She's a recipient of the Kaplan International Prize for advancing understanding of polycystic kidney disease. She's an elected member of the American Association of Physicians of Women in Pediatric Academic Medicine. And she has also served on the NIDDK at NIH Advisory Council. Dr. Guy-Woodford's early studies use mouse genetics in a very powerful way to identify the congenital polycystic kidney locus. She's been involved with developing prenatal diagnostics for polycystic kidney disease, identifying the disruption of a novel ciliary-associated protein in the mouse CPK model for polycystic kidney disease, And if you really want to read an amazing paper, and she's got a bunch of them, and I think she's probably going to talk about it when I saw the title, it's the paper from Kidney International that was published, what, 4 weeks ago, 6 weeks ago, something like that. It's a great paper. I mean, please read it. Dive into that paper, and you'll see how the sort of work and the way Dr. Gay Woodford approaches problems and thinks about it has taken really fundamental animal model, genetics, human disease, and so forth, to really get to understanding a mechanism, in this case of the cystin intracellular trafficking, and how a deficiency in this could lead to disease pathogenesis. And she studied several families with children that have this disease. And she uses in a very elegant way optogenetic methods to study how phosphorylation and dephosphorylation, I know I'm getting in the weeds here, but nonetheless, how that affects this pathway. But to me, it represents the essence of really beautiful science, thinking about a problem, but on the other hand, just like Leslie's first slide, thinking about it in a patient-centric way and what this could do for families that are otherwise dealing with a devastating disorder. So without further ado, please join me in welcoming Dr. Lisa Kenman.
What a lovely introduction. Thank you. So I have known Mike since I was younger than a pup. It's really a pleasure to be here. Thank you, Mike, for the introduction. Thank you, Nikki, there you are, for the invitation. As you know, I was supposed to come, and then Mother Nature wreaked havoc on the DC area, which quite frankly could mean just a few snowflakes in the air, but we actually got 2 feet of snow, so it was really havoc. And I thought, this would be really fun to come and talk to you folks as medical students and as graduate students. And I want to tell you a story. And so I hope that I can tell you an interesting and compelling story in the next 40 to 45 minutes. And then I look forward to hearing your questions about the story and about my journey. So, of course, I have to reveal my potential conflicts of interest at Otsuka Pharmaceuticals and Estuary Biotherapeutics. I serve as an advisor to both companies. So this story unfolds in 3 major parts. First, I want to tell you about the recessive form of polycystic kidney disease. Then I want to tell you about our discovery that a gene called CYS1, which we identified as the causative gene in the CPK mouse model, as Mike spoke to, and has a phenotype that looks for all the world like human ARPKD. Actually, in fact, the human counterpart of this gene is involved in human ARPKD. And then what I wanted to talk with you a little bit about is how, armed with this insight that this is a human disease gene, how did we go about trying to figure out what in the name of God does this gene do? So let's start with an overview of ARPKD, the human disease and mouse models. And I'm going to have, in the course of this talk, a sort of a pas de deux, dancing back and forth between human disease and mouse models. Human disease I deal with every time I go to the clinic. For those of you who are clinicians, that really is a human laboratory, when you go to the clinic and you see patients. And then when you go to the laboratory, it's a much more controlled environment with your experimental models. And for me, that's the mouse. So autosomal recessive polycystic kidney disease has an estimated incidence of 1 in 26,500 live births. That's a figure that we arrived at after a EHR-based analysis and published in 2017. It's a very interesting disease. It's not the soap bubble disease of polycystic kidney disease. It actually involves dilatation of the last portion of the nephron called the collecting duct, and it results histopathologically in these sort of striated, dilated ducts that run perpendicular to the renal capsule up here. It is invariably a kidney disease and a liver disease. The liver disease involves a block in the terminal differentiation of a network that involves the portal vein and its ramifications, and the bile duct lattice-like network that envelops these branching portal veins. That doesn't happen normally. It doesn't complete the developmental process normally. And so you end up with a venous pattern that is, if you will, somewhat pruned, and a very chaotic bile duct configuration associated with these venous ramifications. And that ultimately leads, that vascular epithelial abnormality engenders, for reasons we don't completely understand, a fibrotic response, which we as clinicians recognize as congenital hepatic fibrosis. So in ARPKD, there's the kidney disease and there's the liver disease. And those are the two major phenotypic manifestations of this disorder. So we, working with an international group, identified the major human disease gene for ARPKD. And for a long time, we thought we were done, except for the fact that we had patients that we collected in our clinic and other clinics that didn't have an explanation. And there probably is more complicated explanation than to just say there are other genes involved. An international group published the fact that there are maybe a score of patients who have mutations in a protein called DZIP1L. And then, of course, we had the mouse model, the CPK mouse model, which in the mouse looks like human ARPKD. If we think about these proteins, FPC, which is the protein product of the PKHD1 gene, has several intracellular locations that bespeak its possible functions. So it's associated in the primary apical cilia, this hair-like projection that sticks off the apical surface of epithelial cells. It's associated with the two major protein products, or the two protein products of the major genes involved in the dominant form of polycystic kidney disease. PKD1 and PKD2, the corresponding proteins are polycystin 1 and polycystin 2. In addition, FPC can be found at the transition zone with DZIP1L and in the basal body and co-localizing with cystin. FPC can be found in mitochondria, and like cystin, FPC can be found in the nucleus. And so these localizations give you some clue about function, but it is a very limited clue. So hopefully the story I'm about to tell you will flesh out what we understand. So again, as a clinician, it's important to me that you understand this disease. So on the sort of vertical axis are the major points in terms of clinical presentation. There is the baby, there is the kidney in an older child or adult, and there is the liver. So in prenatal context, the major problem is decreased amniotic fluid, and the related problem, cause and effect is difficult to completely ascertain, but the related problem of pulmonary hypoplasia. In fact, when these children die in the newborn period, and our analysis of the EHR suggests the perinatal mortality rate is about 21%, when they die in the perinatal period, they die of pulmonary hypoplasia. When they are born and they survive the first couple of days of life, their problems can include impaired feeding and growth, respiratory insufficiency, which actually tends to get better, and hyponatremia. Fascinating problem that, if you're interested, I can talk about. For older children, enlarged kidneys is primarily their problem. These are, when you look by ultrasound, they're echogenic. Because ultrasound has gotten so sensitive, sometimes you can see cysts. 10, 15 years ago, you couldn't see cysts, but the level of resolution of ultrasound has gotten better. Decreased kidney function over the course of childhood and into adulthood is a problem. For reasons we don't understand, these children have an increased risk for UTIs. I don't just mean white cells in the urine, which they all have. I mean evidence for urinary tract infection. Systemic hypertension can be a major clinical problem to try to address effectively. And in the liver, the consequences of congenital hepatic fibrosis can lead to portal hypertension, dilated bile ducts, which are a setup for a very particular kind of infection called cholangitis. The bottom line is this can be a devastating disease. So what's the problem? Well, in medicine, when we ask what's the problem, we often turn to experimental models. The problem in ARPKD, and this is just a sort of an annotated list of the mouse models of PKHD1, most of these were engineered, and they whack the gene, right? You have no protein product, apparently, when you make these different models. These mice all have the liver lesion, and very few of them have any kidney disease at all, and that's mostly expressed on a mixed genetic background. On pure genetic backgrounds, they don't have kidney disease. So here you've taken a human gene that, if you tickle in the human, gets overwhelming kidney disease. You can hit it with a sledgehammer. It sounds a little violent, but you can hit it with a sledgehammer in the mouse and you don't see any disease. So that's a bit of a problem for sorting out the pathogenesis. The DZIP1L, as I said, was reported by an international consortium as being the second ARPKD gene. To me, the problem with that is that in the mouse and the human, they never get liver disease. Is this another form of hepatorenal fibrocystic disease, or is it really ARPKD? I would argue it's the former, not the latter. And then you have the CPK mouse. And until very recently, we didn't have any human patients with CPK, so we didn't have a model to really study. When we think about human patients, most patients have the kidney disease and the liver disease. There is a small set of patients who have only the liver disease. Fascinating group of patients. So they have no structural or functional abnormalities in their kidneys. And then, as I mentioned, the DZIP1L patients have the kidney disease, early onset kidney disease, but they don't have any liver disease. So just comparing the kidney and the liver, human and mouse, you can see the mouse is really remarkably similar to the human in terms of histopathology. This is the kidney histopathology. This is the liver histopathology. Another thing that I need to tell you so that you understand the sort of the threads of the story is that for a long time, in work that has been primarily championed by Marie Trudel in Montreal, we know that MYC is overexpressed in renal cystic epithelia. So using immunohistochemistry, we've shown that it's overexpressed in the epithelia of the CPK mouse. When we look at protein expression, it's 10, the c-MYC protein is 10-fold higher in CPK kidneys, and the mRNA is 2-fold higher. And when we look at human ARPKD, and this is one of the first studies to show this, this is a series of patients that either are compound heterozygotes for 2 truncating mutations, that's A1 through A3, compound heterozygotes for 2 missense changes, that's A4, or compound heterozygotes for a missense change and a truncating change. And you can see for all of them, the level of MYC is elevated. So overexpressed MYC is a signature not just of the dominant form of PKD, which Trudel reported, but also the recessive form in both the human disease and the mouse model. So one of the big insights in terms of the importance of the CPK mouse model was having a human patient that actually had mutations in the human counterpart of the CYS1 gene. And so this is the first clinical case that we reported in collaboration with Friedhelm Hildebrandt in 2021. So it was a 5-year-old boy born from a consanguineous union who presented with polyuria, polydipsia, and poor growth. His renal ultrasound revealed that he had multiple medullary and cortical cysts. Remember, I talked about the sensitivity of ultrasound. You can even see a few cysts. Because he was consanguineous, we could apply the tools of homozygosity mapping. We're looking for regions where across the genome where there are regions of homozygosity, ROH, okay? And the red spikes are those regions of homozygosity. What caught our eye was there was a region of homozygosity on chromosome 2. And that's where the CYS1 gene sits. And so we did a whole series of procedures. The bottom line is we got his parents and him, and we found that he was homozygous for a splice site variant. So here, the splice site between exon 1 and exon 2. Just for your reference, the CPK deletion is here at the tail end of exon 1. So this was the first demonstration of a human patient that had CYS1 mutations. And what were we to make of it? Well, we did a lot of work, but in relatively short order, we were presented with another child. Again, consanguinity of a 5-generation pedigree shown here on the right. Day 1 of life, very large kidneys, poor corticomedullary junction, differentiation rather, and the liver was anatomically normal. And you could say, no, wait a minute, you told us that liver disease is an invariant part of ARPKD. It is histopathologically, but in very young children, the sensitivity of ultrasound isn't deep enough to pick up those abnormalities, right? So it did not surprise us in a neonate that he had a normal liver, at least anatomically. And so my colleagues in the UK, their standard approach for genetic analysis was first to do array CGH. And they found there we go, a deletion here. It's a 24 megabase deletion that takes out the first exon of CYS1 and the regulatory region. Okay, and he's homozygous for this. So this kiddo is predicted to not make any CYS1 protein. So now we have 2 kids, 2 entirely different families who don't make any CYS protein. Now it's really kind of the impetus to sort of figure out what does this protein do and why, missing this protein, do you get ARPKD? So we went back to the mouse. The first thing is this gene expressed in the right place? And so we decided to use in situ to have have the ability to look in the developing mouse. And so if you start on the left side, you go left to right in increasing levels of magnitude, and you can see in the kidney, the expression is in ductal epithelia. The arrow here points to the developing liver, and again, the expression is in ductal epithelia. Makes sense with what we know about the human disease. If you're a little bit later in the developmental process, so this is post-day conception 14, this is post-day conception 16, again, it's ductal epithelia. So the protein's in the right place in the normal mouse. We went on with Brad Yoder right around the time that PKD proteins were found to be associated with the cilia. We made the requisite polyclonal antibody. We looked in the developing kidney and the postnatal kidney, and the arrows, for a clue, point out where the cystin protein co-localizes with a marker for primary apical cilia. And then when we look in cell culture in the XY or the XZ axis, again, it co-localizes with markers of the primary cilia. So cystin is a cilia-associated protein. But how does it get to the cilia? Well, we took advantage of the fact that the HIV GAG protein, the first 30 or so amino acids, has the ability to associate with the membrane through a myristylation signal. Well, we thought that's great because, as you'll see in a minute, cystin has a myristylation signal. So we made a series of constructs that were basically progressive chopping off the carboxy terminus of cystin until we got to one where we chopped off 110 amino acids. So it only had 7 or 8 amino acids that were additional to the CPK protein that would be predicted. And when we hook that up with the HIV GAG protein as a chimeric protein, it's expressed in the primary apical cilia. That allowed us to say, because we knew the CPK protein's not expressed in cilia, that allowed us to say that this region likely had a cilia targeting signal. And when you look over the sort of phylogeny, it is a highly conserved signature for cystin, but it doesn't have any bearing to other cilia targeting signals. Precisely to make the point that there are signals for proteins to be targeted to subcellular compartments, but it's not universally one signal. So you have to kind of chase that down in terms of true cell biology experiments. We went on to look at the protein informatically, and we saw, oh, it has 2 nuclear localization signals. Could it not only target to the cilia, but maybe it also goes to the nucleus? And if it does, which one of these or are both of them required? So again, using a series of constructs, we were able to show in fractionation studies, and I'm just going to go immediately in the interest of time to the immunofluorescence study. So here's the full-length protein, and you can see it decorates the membrane in this image. If we take off the first 20 amino acids for this protein, it is in the cytoplasm. It's not really decorating the membrane, but it seems to be excluded from the nucleus. Whereas if we take off the myristylation signal and we have this nuclear localization, the most N-terminal nuclear localization signal, this protein almost, at least in immunofluorescence, goes exclusively to the nucleus. So that first nuclear localization signal here is a functional nuclear localization signal. So now we have a cilia targeting signal, can send it one way, and a nuclear targeting signal can send it another way. That's all really lovely, but what is it doing in those compartments? So we first tackled the nuclear compartment because we were interested in transcriptional regulation and what are the genes that this protein might be involved in. And if you dysregulate this protein and therefore dysregulate the expression of these genes, is this part of the signature of polycystic kidney disease? So we did a yeast-2 hybrid screen, which many of you may know nothing about. And that's perfectly fine. We identified 20 potential interacting partners. We confirmed interaction for a number of them with the classic methodologies, and we came up with nectin. The problem with the nectin protein is it's an imprinted protein, and to that point, it had been primarily described in post-mitotic neurons. A little bit in adipocytes and a little bit in islet cells, a little bit of information in that regard, but not a whole lot outside of the nervous system. In terms of functions, it seemed to be a DNA protein binding protein that bound to the GN box of the MYC promoter. Remember, I talked about MYC is important in this disease. And it interacted with other transcription factors in this regulatory process. So we said, oh, this is very interesting. So here's again, just to remind you that MYC is overexpressed in the CPK mutant. To just drill into to the regulation of MYC expression, MYC has two promoters, the P1 and the P2 promoter. The P1 promoter is driven by a GN box. So we said, great, we'll take this section and we'll clone it into a luciferase vector and use luciferase activity to help us determine what's driving what. And when we do these luciferase assays, you can see if everything is normalized to 1, that nectin increased luciferase activity. And that if you put in cystin, that was decreased relative to this peak. But if you put in cystin that was missing the last 25 amino acids, it was the same as nectin. You could ask, well, why? Well, it turns out in that last 25 amino acids of cystin is the binding site for nectin. So the reason it was like this is because it couldn't bind nectin, so it couldn't interact. But we had a little problem because no one had described that nectin was an epithelial protein. And so, we created stable cell lines expressing cystin GFP and nectin RFP. And we looked, and you can see nectin is beautifully in the nucleus. That's the red staining here. But cystin, not so much. We said, aha, maybe because cystin is moving in and out of the nucleus. So we did a little trick, a little cell biology trick, and we blocked proteins from getting out of the nucleus. We're like, we'll trap it and we'll see if it's there. And sure enough, with the trap, it's there and it co-localizes with nectin. So now in the right kind of cells, epithelial cells, renal epithelial cells, we have cystin and nectin both in the nucleus. So as an interval summary cystin functional insight, it's a cilia-associated protein. It turns out other folks working on similar proteins, similar N-myristylated proteins, were able to show that cystin and the major protein, one of the major proteins in a disease called nephronophthisis, are transited from the endoplasmic reticulum to the cilia by a chaperone called UNC119B. When it gets into the cilia, RL3, which is regulated between its ATP and ADP state by RL13B. The RL3B that's GTP bound releases UNC119 from cystin, and cystin associates with the membrane. Then UNC119 traffics out of the cilia, and RL3B, the GTP bound state, remains in the cilia. So there's at least a way in for cystin. So that was important. In addition, we have evidence that cystin interacts with nectin as a transcriptional regulator. And what's interesting, it ties in very nicely with work that had been done by the Kansas group, saying that MYC antisense oligonucleotide treatment ameliorated the cystic kidney disease in the CPK mouse. Okay? Well, that's all well and good, but there's still a whole lot of black boxes here. Like, what is cystin doing in the cilia? Does it move from the cilia? How does it, is there a regulated pathway into the nucleus? And what's required for that? And we got our insight with the third family, which showed a functional loss of the putative N-myristylation site in cystin 1. And they had the siblings in this particular family had an ARPKD phenotype. So successive fetal siblings, these were actually children that were, at CHOP, we have something called a special delivery unit for high-risk pregnancies. And each of these children, their successive siblings, were delivered at CHOP. They had the oligohydramnios that you see in ARPKD. They had pulmonary hypoplasia. The first child died at 1 day of age. The family refused any post-mortem evaluation. Fetus B came along, looked like it had the same disease. The family not willing to go through the sort of the pain and the incredible odyssey, actually had that pregnancy terminated. When we did exome sequence analysis of the affected fetuses and their parents, we found that both fetuses, both children, were homozygous for a missense change that converted the glycine in the second position to a serine. And we looked at all the other, and there were more than 100 genes that are associated with renal cystic disease. We looked at all the rest of them, and none of them had any variants. So based on this data, these data, and the data from our 2 previous families, we said this is probably the disease-causing change. And it was important because we knew cystin had a putative N myristylation signal. So we generated a web logo schema. And for those of you who are interested in this, I'm happy to talk about it. Basically, it looks as the strength of conservation across a protein when you look phylogenetically. So 97 mammalian cystin sequences were put into this analysis. You can see the myristylation site is highly conserved, but and the myristylation signal, because N-terminal myristylation is an enzymatic reaction, is all highly conserved. And then there's this polybasic domain that's highly conserved. And then, of course, the cilia targeting signal. What we know about N-myristylation comes largely from work that was done on the Marx family of proteins. And what those investigators were able to show is that there's such a thing as membrane association. N-myristylation has an enzymatic reaction that takes myristotic acid, which is a 14 carbon acyl fatty acid, and through an enzymatic reaction, sticks it onto the G2 glycine in a protein. That is necessary but not sufficient for membrane association. So you either need another acyl group, like a palmityl group or a farnesyl group, to be on the protein to secure it in the membrane, or you need a polybasic domain. We had a polybasic domain. And it's interesting because further evidence suggested that these non-membrane associated proteins were stabilized by these N-myristylated proteins. And among them was PKA, and that will be important in a minute. So one of the things that we did was there's a way in terms of fractionation studies to say, oh, this is probably a functional N-myristylation signal. And N-myristylated proteins separate in fractionation studies in certain fractions because these are lipid microdomains. They have more cholesterol, more gangliosides, and more sphingomyelin. And so in the membrane, they make sort of a liquid ordered domain. Whereas in the other portions of the membrane is very fluid because it's mostly phospholipid that have unsaturated acyl groups, okay? So the membrane's fluid except for these liquid ordered domains, which would make sense if these liquid ordered domains are signaling platforms. And we were able to demonstrate that the wild type protein is associated with lipid raft microdomains. We then went on to demonstrate that N-myristylation and a novel cilia targeting signal are both required for cilia localization. So here's the wild type protein. Here's the CPK protein. It's got the N-myristylation signal, but it's missing the cilia localization signal, as you can see right here. And it doesn't associate with cilia. And then if you disrupt the N-myristylation signal, like in our patient, it also doesn't associate with the membrane. So both of them are necessary. And so this brought us to the fact that this is probably cystin is a membrane associated with a putative myristyl electrostatic switch. Here's the model in terms of the sequence and the model in terms of the membrane. So there are positively charged amino acids that are separated by serines, and these serines can be phosphorylated, decreasing the affinity for the membrane and releasing the protein from the membrane. And when we look at the cystin protein, there were 2 strong peaks for predicted phosphorylation at the serine in the 8th position and the serine in the 17th position. So we said, okay, we're going to make an anti-phospho antibody. So we sent to a company, said, phosphorylate this serine, send it back to us, and we're going to make a monoclonal antibody. We did that. And you can see that the only thing that was recognized by this monoclonal antibody was the phosphorylated peptide. So they fixed the phosphorylation state on this peptide. Important because it's an important tool. And when we use this phosphorylated protein and examined where is cystin that's phosphorylated, it's excluded from the cilium. Saying you phosphorylate cystin at S17 and it traffics out of the cilium. So you can't find it there. Okay? So this is an important point. We then said, okay, that's all the mouse. what happens in the human? So we used mouse cells because we're very familiar with these mouse cells, and they're collecting duct cells, terminally differentiated. And we put in the human wild type cystin and the cystin that was predicted to be transcribed in our human family with the G to S substitution. And what you can see over here is the wild type protein did very nicely going to the cilia. You can see that here, and you can see that here. Whereas with the G to A substitution, it couldn't find its way to the cilia. Okay, so maybe that's the problem with this, with the protein in these families. That's great, but what role does that have to play in cystic disease? So everybody uses Cas-CRISPR now, so we used Cas-CRISPR. The problem with the CPK gene is that the first exon is incredibly GC rich. So have any of you used CRISPR-Cas technology in your work? Thank you. I see two hands behind the computer screen, and I appreciate your response. We just couldn't target this G because there were all these, this glycine, because there were all these guanines around it that interfered with our guide RNAs and homologous DNA repair. So we said, okay, well, we have all of these constructs in these different lines where we've completely disrupted cystin. So this C1 line, you can see we have no reads of the wild type protein, reads of the first variant here, where we had a 2 base pair deletion, probably in the Kozak sequence. So we predicted it wouldn't make protein. I'm not showing you the immunoblot here, but it doesn't make protein. And then here, where we had a 7 base pair deletion that takes out not just the part of the Kozak sequence, but also the ATG. That also doesn't make protein. So functionally, this is a cystin null cell line. And we said, great. Now we're going to put in the human constructs, the wild type and the G to A. And you can see the wild type, in fact, is beautifully expressed in the cilia, and there's no cilia expression with the G to S. Now, there's a little trick in renal epithelial biology that if you grow cells in Matrigel and you treat them with forskolin, activating cyclic AMP, they will become cystic. And so it wasn't a question of cystic, non-cystic, it was the extent of cystic change. So if a cell is null for cystin, you could see that there was cysts formed, both in terms of number and in terms of area. If you put in the wild type, there were very few cysts that were formed despite forskolin treatment. If you did the G to S, there was very exuberant cyst formation, both in number and in area, and quite frankly, more extensive than just in the null cell alone. So we now had a connection between a phenotype of cystic disease and this G to A transition. So we'd say, oh, well, the problem for this particular family is that the myristyl electrostatic switch in cystin can't work because the cystin never gets to the cilia. That's part of it. But then what's phosphorylating the cystin was a question to be asked, because that would determine how we track this down. Well, I mentioned PKA. PKA is kind of the canonical, one of the canonical cyclic AMP pathways. And we demonstrated, and I'll just cut to the chase here, that cystin and the R1, one of the major subunits of PKA, co-localize in cilia. That put them in the right place. If you had wild type cells, cystin, of course, is expressed in the cilia. If you treated these cells with 8-bromocyclic AMP, again, to stimulate cyclic AMP signaling, the level of cystin in the cilia went down. So we thought we had a PKA-mediated cyclic AMP process that could phosphorylate cystin and lead to its trafficking out of the cilia. But how could we prove this? A cilia is about anywhere from 20 to 100 microns long and just a couple of microns wide. And it is a protected compartment in the cell. How could we use our blunt tools to be able to get at what's going on there? So we turned to a colleague in Germany who is a whiz at optogenetics. And what Dagmar has in her portfolio of clever tools is she has a photoactivatable adenylate cyclase. Okay, adenylate cyclase is what generates cyclic AMP. And because of the tag, remember this NPH3 protein that's an N-merystylated protein that travels to cilia? So she has a construct that has NPH3, a Florenza tag, M-cherry, and she calls that the cilia version of her BPAC. And then she just has BPAC and M-cherry that's in the cytosol. And light will activate this cyclic AMP, so will forskolin. So in the dark, it's in a resting state. If you expose it to light or forskolin, you activate it. And this allowed us to separate phosphorylation in the ciliary compartment from in the cell body. So actually, this projects better than I thought it would. The top panels are dark. The bottom panels are light. I'm sorry it's so small, and it was small in the paper too. Let me cut to the chase with the red boxes. If you look at cells that where you're looking at the ciliary BPAC, the wild type, the wild type cystin in cilia is lower, statistically significantly lower than it is in the cytoplasm. Okay, whereas if you just look at the wild type, there's no difference. That doesn't have the BPAC. There's no difference between them. If you go over here to the phosphorylated antibody, so we're only looking at phosphocystin, what you see is all of a sudden the amount of cystin in the nucleus and in the cytoplasm is higher than at baseline. How could that be? Well, think about it. If the amount of cystin in the cell doesn't change and you decrease it in the cilia, where is it going to go? There's no back door to get out of. So it will increase in the cytosol. And because it also traffics to the nucleus, we would not be surprised to also see it be higher in the nucleus. So this was a wonderful proof of concept. When we then looked at pKa, we could see that both cystin travels out of the cilia when stimulated with this adenylate cyclase, as does the pKa isoform. Okay, so these data taken together indicate that cystin has a functional myristoelectrostatic switch. So one of the things we were very interested to, just the last sort of portion of the discussion, was, okay, so cystin's coming out of the cilia, it goes to the nucleus. What does it do there? It seems to be important in regulating nectin. Does it regulate other genes? And does it need partners to make this trafficking journey and to regulate the transcription of other genes? So here we used a affinity purification method called TAP. So again, to just remind you, there's wild type cystin that decorates the membrane, and the G to A, which is increased in its localization in the nucleus. We were very interested in cystin's binding partner in the nucleus. So we used the G to A version. We did 4 different experiments, and we came up with 80 proteins that we found they were common to every single experiment. And interestingly, one of those proteins, so there were 2 members of a family of serine threonine phosphatases, PPM1A and PPM1B. They're very closely related. They have both non-redundant and redundant roles in cells. What's interesting is they're both N-merystylated. So in membranes, it could put them in the same place. And so we looked to see if we could find a physical interaction between the two, because if this is an enzyme, does it bind to the protein that it's modulating in terms of dephosphorylation? But you don't capture those sorts of things with our standard tools. And so we used a physiologic approach. If we take a specific inhibitor to PPM1A, which is sanguineurin, can we decrease or inhibit cystin S17 dephosphorylation? And you can see in a dose-dependent fashion, we can. So that allowed us to come up with the following model. In the wild-type situation, cystin mRNA is translated. It has the G2 domain, which is merystylated. It therefore traffics probably under the sort of the pathway that involves UNC119B into the cilia. It's released into the cilia membrane by the action of RL3B, and it sits there. the cilia is stimulated by something. We still don't know what that something is. And that activates the cilia-dependent adenylate cyclase, increasing cyclic AMP in the cilia, phosphorylating the S17, and cystin traffics out of the cilia, makes its way through the cytoplasm into the nucleus, where it binds with nectin and regulates at least c-Myc transcription. In the case of the substitution at the second glycine, either with an alanine or with a serine, cystin never gets to the cilia. Remember, I talked about these lipid microdomains are signaling platforms. Lots of proteins associate with one another there. Our speculation is cystin's got to go to cilia to pick up its buddies, its functional buddies. If it doesn't get there, it doesn't have its functional buddies. And so it can make its way in the cytoplasm, it can make its way in the nucleus, but it doesn't have its buddies for proper transcriptional regulation. And so therefore, MYC expression is not regulated in the way that it should be. That's kind of captured in this schema on the right-hand side of the diagram. So part of it is the regulation of cystic trafficking through this myristoyl electrostatic switch, but part of it is the cilia localization in this special compartment in the cell probably provides cystin with binding partners that it wouldn't otherwise pick up in the cytoplasm. And those binding partners are probably very important in its nuclear function. So to conclude, based on our data identifying 3 independent proteins with an ARPKD phenotype and homozygous CYS1 variants, ClinVar is now recommending that CYS1 get added to the renal cystic disease gene panels that we use clinically. In our functional characterization, primarily of mouse system, but in a spot way, checking also the human protein, both N-myristylation and a novel targeting signal are required for cilia localization. Cystin intracellular trafficking is likely differentially regulated spatially and temporally in specific subcellular compartments, what I was just talking about. When it reaches the nucleus, it acts as a transcriptional co-regulator, probably working in a protein complex that still needs to be defined, modulating the expression of MYC and perhaps additional genes. And I think that in the absence of a mouse model for PKHD1 that faithfully recapitulates the human model, this provides us with a model to begin to understand the ciliar nuclear interplay that's critically involved in the pathogenesis of this disease and the role of these sister proteins in regulating genes like MYC that are really involved in driving the cystic phenotype. So let me stop there and acknowledge, for the purpose of this project, my CHOP team and collaborators, my Children's National team and collaborators, US collaborators, international collaborators, and my funding. And in the last few minutes, I'd be happy to take your questions. Thank you for your attention. Thank you for the wonderful talk. Dr. Friedlander.
So I couldn't resist, as a neuroscientist, thinking about ciliary to nuclear communication, thinking about neurons and dendrites and local protein synthesis and dendrites way out here and communication back and forth. So I was just wondering, from a therapeutic intervention point of view, and I was thinking some analogies in brain disorders, but anyway, from a therapeutic intervention point of view, what is your best plan or hope or strategy, as you would like to characterize it, to where in that process is most likely to be targetable or druggable, I should say?
So we actually have just worked in collaboration with a bioengineer at USC. Yunji is very interested in delivery systems to different cell types. And in this experiment, we used the CPK mouse, collected urine from these mice. And it turns out urine sheds not only liquid, but also exosomes. These exosomes have very interesting, not just the proteins, cystin, polycystin 1, polycystin 2, a portion of the FPC protein, but it also, the KIF12, which you mentioned, is a modifier gene for ARPKD. But it also has mRNA as well. And what we were able to show is in taking these urinary exosomes from the urine of normal mice, we could introduce these exosomes into the circulatory system of mice that have a CYS1 defect. Not correct the defect, but attenuate the progression of the disease such that mice live twice as long. Now, these mice don't live a long time. That wasn't that hard a task. But instead of, a couple of weeks, they lived several weeks. So it's a proof of concept. So one thing is to say, just put the normal protein in and the cilia will figure it out and it will be released from the cilia in a regulated fashion and the world will be a better place. That's one approach. So you folks know I'm from CHOP. Implicit in Mike's question, because there's always multiple questions implicit in Mike's questions. Implicit in Mike's question is, well, why can't you baby KJ this, right? Well, the problem is, remember, baby KJ had a urea cycle defect, okay? Urea cycle defects involve the urea cycle, which is primarily in hepatocytes. Hepatocytes are very amenable to targeted therapies. And they're also very amenable to taking up vectors like the CRISPR vector that allowed the folks at CHOP to correct the defect. The kidney is not as amenable to vectors, certainly AAV vectors. People have been working this a long time. Maybe a little bit more to LNP vectors. Maybe a little bit more to the vectors that can put ASOs into cells. But we have a proof of concept that taking a different approach and exosomes, that renal epithelial will take up these exosomes.
Hello.
Hello.
Thank you for your talk. Just some context about me. I'm not a medical student. I'm a grad student, mostly working in the brain. So I don't know very much at all about the kidney. But I'm just curious. It seems like the cilia is really critical. And you had mentioned that, the cystin goes to the cilia and it sits there and it's waiting for something to happen. And then it gets phosphorylated, comes back, and it does its job and regulates transcription. Do you have any hunches about what that stimulating factor might be?
Could be a number of things. It's a great question. So first of all, it's not just sitting there. It's probably in a regulatory platform making associations with other proteins, particularly other N-myristylated proteins. So that's the first point. The second point is that work started by Ken Spring back in the late 1990s and progressing through a series of really elegant studies from labs around the world demonstrate that the cilia is an antenna. It's a sensor. It seems to be not only a mechanosensor sensing flow, but it also seems to be a chemosensor. There are also studies that have suggested that it may be part, as a neuroscientist, this is probably familiar to you, it may be part of sort of an extracellular cyclic AMP pathway where cyclic AMP gets converted to adenosine and adenosine binds to its receptor. I mean, there are all kinds of sort of different processes. So I think that what we can say is the following, that all cell types of the body have a primary cilia, except for 4: circulating blood cells, adipocytes, hepatocytes, and there's some debate about this last one, intercalated cells of the collecting duct. Everything else has a cilia. Now, in cells that are not polarized, that it's not the same sort of distribution. But for example, Jennifer Lippincott-Schwartz, who is a major cell biologist, as you probably know, and does these most exquisite imaging sorts of studies, has shown cells that can sit down next to an adjacent cell with its cilia tucked into a pocket, sensing what that other cell is doing, right? So I think that the sensory nature of cilia is really important, and it triggers a bunch of pathways that involve either the movement of proteins in and out of the cilia, the proteolytic cleavage of proteins that are in the cilia. For example, FPC undergoes notch-like processing, releasing the N-terminus into the extracellular fluid, into the urine space, and the cytosolic tail into the cilia space, and it traffics out of the cilia. What all these signals are, we don't understand, but the signals probably change in development. Because remember, there isn't urinary flow in the kidney in the developing kidney. So in fact, Lisa Satlin's group, now maybe 25 years ago, showed this beautiful image of a rabbit nephron, a distal nephron, where the cilia are reaching across the lumen. The flow is very low, and they're touching one another. And so there may be, you know, a physical kind of interaction between these cells across this potential space via their cilia. Does that answer your question?
Yeah. I do have a follow-up question.
I'm not so sure. Why don't you go ahead and ask?
Well, so my follow-up question would be that for the most part, every cell body has the cilia. So why is it that if this problem is really sort of component of the kidney?
It's a great question. It has to do with every cell of the body may have a structure. Does it have all of the protein apparatus? You know, until our work showing that nectin was in renal epithelial cells, nectin was thought to be a post-mitotic neuronal protein. And it's only when people have started to look. Why? Think about it another way. Think about it from a genetic point of view. If every cell of the body has the genetic defect that you see in either an autosomal dominant or an autosomal recessive, let's take autosomal recessive. Why do we only have kidney and liver disease? Right? And so that you know, part of what makes our biology special, I mean, we don't have that many more genes than a worm, right? But it's the way they are transcribed in a temporal and spatial sort of way. It's the isoforms, you know, many genes, maybe 40 to 60% of genes have multiple splice isoforms. It's the way that partners in pathways are there or not there, right? So I think your question is it's a fundamental question in biology. Why is there organ specification? And it probably has to do with the cassettes of proteins that are involved in pathways, either being wholly represented or not. I mean, that's the idea of repurposing drugs, right? So for example, to push this PKD analogy, you know that tolvaptan has been approved for the treatment of patients with the dominant form of disease, and it being very severe and really very aggressive. Tolvaptan was designed for people with heart failure because it blocks the V2 receptor, okay? But that cassette is there, obviously, in renal epithelia. And it turns out cyclic AMP is a major driver of cystogenesis. So the cassette was there. We just needed to think about it differently. Sure. Thomas.
Hi, from up top. So correct me if I missed something, but it sounds as if in your mouse models, the majority of your research has focused on the renal ductal cells, but we talk about the hepatic association. Has there been any study or theory involving the role in renal cells, involving MYC or other regulation?
Yeah, so not as much. Part of the reason is we have laboratory tractable tools for the kidney more than we do for the liver. So there aren't lots of immortalized cell lines that are easily available for cholangiocytes, right? Nick LaRusso's lab at the Mayo Clinic produced those. And they have done a lot of work, but they've been working not in the CPK mouse, not in human tissue, but in the PCY rat. The PCY rat is the rat ortholog of PKHD1, and it is a splice abnormality. And it gets both kidney disease, but it's much more slowly progressive, but it gets the full bore ductal plate malformation, and that's where the work has been done. Have they looked, because it's a great question, have they looked to see if cyclic AMP, yes, they have looked at cyclic AMP. Have they looked at MYC as an effector? One of the problems is you don't see cis so much in the liver. What you see is basically a block in terminal differentiation. So think about it this way, Thomas. If you had a bush that had this beautiful lattice of very fine vines growing up around the ramifications of the bush, that's the normal biliary tree around the vascular ramifications. If the bush gets sick and the branches get pruned, and something bizarre happens with the vine, so it's now thick and it's chaotic, that's what you see. So it's a very different pattern. And understanding is a block in terminal differentiation, what's going on in the kidney and in the liver. It's possible that they're related, but our ways of sort of sorting that through are a little bit different. And the interest in this has been primarily Nick LaRussa's lab at the Mayo. So good question. The information's incomplete.
All right. Thank you so much.
Sure.
Wonderful talk.